The eLife podcast from eLife, the open access journal for outstanding research in the life and biomedical sciences. Online at elifesciences.org.
Hello, welcome to episode 49 of the eLife podcast, which is produced by The Naked Scientists. I'm Chris Smith. This month, the genes that give pigeons their plumage patterns, why the EU's demands for bovine TB vaccination trials seem totally impractical, and why we've been measuring the wrong thing in patients with lung fibrosis. But first, arguably one of the most important questions in biology is how life got started on Earth in the first place. A popular theory is that it began with DNA's single-stranded molecular relative, RNA. RNA can carry genetic information, but it's also capable of folding itself into complex three-dimensional shapes that also endow it with enzymic or catalytic capabilities. The problem is how to make a tangled 3D structure like this copy itself. DNA and modern RNA systems do it by using proteins to temporarily straighten out the strands to make them readable. But early life wouldn't have had access to those proteins. So how did it do it? Phil Holliger.
So in present-day biology, DNA stores information in the nucleus, proteins is the sort of bricks and mortar and the engine of the cell, and RNA is like a carrier of information from the information stored to the machinery, to the proteins. But I think there's compelling evidence to suggest that early life must have been a lot simpler. So there must have been a primordial biology which may have been based mainly on RNA. And that really raises the question how these RNA strands were copied in that primordial biology, because in present biology, both RNA and DNA strands are copied by proteins. So in that early biology, RNA would have had to be able to copy itself.
There is a precedent for RNA having enzymic and catalytic function, though, isn't there? Because there are things like ribozymes, which are machines in cells where they exploit the fact that RNA has a structure and it can guide chemical reactions. So there is a sort of precedent for having that view.
Exactly. So this is actually one of the reasons why people think that early biology might have been based on RNA. That unlike DNA, which is more like an informational string as a molecule, RNA has this amazing capacity to both be an informational string and fold up into intricate three-dimensional shapes which can have catalytic function, including the capacity to actually copy other RNA strands.
So what's the problem with the hypothesis then? If RNA can do that, it can be both informational and it can be biologically active as a catalyst. Why couldn't early life have just used that?
When we were trying to build such a system where RNA could copy itself, we hit upon a snag. The ribozyme, although it could copy RNA strands very well if they were unfolded, if they were strings, once they had assumed a three-dimensional shape, they could no longer be copied. So you essentially hit a paradox where RNA needs to fold up into a three-dimensional shape to be able to copy itself, but once it has folded up, it can no longer be copied. The paper is about how we have begun to find a solution to this paradox.
How did you?
The idea was the following, so that maybe what we needed to do is to move away from trying to build something that was closely analogous to present-day biology. In modern-day biology, when DNA and RNA are copied, they're copied in single letter steps. So single DNA or RNA letters kind of are incorporated one by the copying enzymes. But and our original ribozyme had exactly the same type of function like this. It would copy RNA in these single letter steps. But then when it hit a structure, it could not move forward. So the solution we found is that if we move to a three-letter step, a triplet as we call it, and this is not used in nature at all to copy either DNA or RNA, when we move to triplets, those triplets, by virtue of binding much tighter to the RNA strand, they could actually straighten out the RNA template, and as we approach the structure, begin to invade and unravel the structures. And this way, we found that eventually we could copy even extremely stable RNA structures, structures that have a melting temperature close to the boiling point of water.
So to put this another way, it's almost like you've got a sort of a shirt with lots of wrinkles in it, and it goes through the iron, gets flattened out.
Yeah.
While it's momentarily flattened out, your enzyme can read that nice flat surface and make a parallel equivalent or a copy of that bit of the shirt, and then it falls off the other side of the ironing board and gets all its wrinkles back again, so it goes back to its three-dimensional shape, but you've got a copy in the meantime.
Well, it's approximately right. Unfortunately, it doesn't actually wrinkle back. I think it will stay as a double strand because that's probably the most stable form. So in fact, this is one of the problems that we are tackling now. To set the system back to its beginning, we will now have to find a way to unravel that double strand again.
You solve one problem and create another.
Well, I think that problem was always there. I think that we always were aware that that was part of the replication cycle. Now in the cell, kind of like there are some intricate molecular machines which have exactly that function to unzip double strands. We will have to find an alternative solution, but we're working on it.
Does this mean, though, that in Darwin's little pool, where there must have been these early forms of life, if they were using RNA the way you speculate they were, that the supply of molecules that they must have had to feed their RNA replication machinery must have been these triplets, three letters joined together in a sequence?
I think the most likely scenario is that, there was a whole, mix of letters there. There were probably single letters, double letters, triple letters, and maybe some quadruple, quintuple, etc. letters. Presumably the longer, more complicated letters were rarer. Presumably the copying enzyme would use whatever's appropriate. So, in a nice kind of extended strand, it could probably quite happily use the single letters, but then once it hits a structure, the more extended letters, the triplets, etc., kind of become more useful.
How did you make the discovery in the first place, though? Was this just brute force, trying loads and loads of variants of the RNA system until it worked?
We do not have currently the molecular understanding of RNA to do bespoke engineering of, let's say, a designed catalytic activity of copying function of exactly that we want. So what we use is an evolutionary process where we force the RNA to begin to use the substrates that we like it to use, like the triplets, by iterative cycles of evolution in the test tube. And this is a very, very powerful process.
So if you've got this right, what are the implications for early life about 4 billion years ago or so?
Potentially there is really no conceptual problems with building such a system from scratch that could copy itself. And with self-replication, presumably the copying process would make a few errors, so you have mutation as well. And with self-replication and mutation, you would have evolution. And that would really get the ball rolling towards ever more efficient self-replication. And you could see how that process would take off and lead to more and more complex things.
A really fascinating hypothesis for what could have powered early life. Phil Holliger, he's at the MRC Laboratory of Molecular Biology in Cambridge. Now, on the subject of evolution, from the University of Utah, Mike Shapiro.
We were interested in trying to understand the DNA level changes that lead to animal pigment patterning. We've known for a long time the types of genes that control the type of pigments that are generated, blacks versus browns versus reds versus yellows. But an outstanding mystery in pigmentation genetics is how those pigments know where to go and how patterns are generated, spots and stripes and so forth.
In terms of actually how the pigments are added to the skin, or in the case of birds' feathers, how is that achieved?
There are specialized cells that are called melanocytes that produce the pigment granules, and those granules are exported onto the surface of either the skin or a feather. There is a fair bit known about the recipe for the granules themselves, but where those granules go has remained a mystery for quite a while. And now, by taking advantage of things that are not laboratory mice, we're able to take advantage of evolutionary diversity to try to answer those questions.
So what have you done?
We focused on pigeons as a model to try to understand how pigment patterns are generated. And in particular, in pigeons, there are a few basic color pattern types. And we've known for a long time, mostly through the work of pigeon hobbyists, that the genetic basis of this is relatively simple. That is, very few genes, possibly even one, have a major effect on how stripes are laid down versus a pattern that we call checkers because it resembles a checkerboard.
So you're saying that people who have bred pigeons over decades knew how to breed for certain traits, but they had no idea what the molecular genetics of this was?
That's correct, and I would extend that beyond decades to millennia. Pigeons were domesticated probably 5,000 years ago, and people have been raising them in captivity ever since. So we know from the long history of pigeon breeding a lot about the number of genes that go into making certain traits. But as you pointed out, we know very little about the molecules involved.
So how did you track them down?
One way that geneticists track down genes that are responsible for particular traits or particular diseases is to identify groups with and without the trait that we're interested in. So in the case of pigeons, the ancestral color pattern is a pattern called bar. It's basically two stripes on the wing. And we compared the genomes of birds that have that bar pattern to pigeons that have what we call a checker pattern, where there's more pigment on the wings in sort of a checkerboard-like pattern. And we tried to identify what was different about their DNA sequences. And when we did that, we identified just one place in the entire genome in over 1 billion base pairs of DNA that was different between those two groups.
And in what way is it different?
When we scanned through that region of the genome, looking for mutations that might affect places that are coding for protein, we didn't find anything that was consistently associated with having a bar or a checker pattern. However, the mutations around those genes were indeed consistently associated. There was one gene in particular that piqued our interest. It's a gene called NDP. It stands for Norrie disease protein. And even though the gene itself wasn't different between the birds with the different patterns, the region very close to it was very different between the birds that had checkers and bars. And through some follow-up experiments, what we found is that that region was associated with differences in the way the gene was expressed. So rather than changing the gene itself, what appears to have happened in these pigeons is the way that gene is controlled, the way its expression is regulated, is different. In addition to that, this same region that differed between the bar and the checkered birds was duplicated in some of the birds with darker color patterns. So it appeared not only that this region differentiated the checker birds from the bar birds, but the more copies of DNA that the checker birds had in this region, the darker they were.
So that's like a gene dosage effect.
We think it's something like that. And again, it's not the gene itself, but when we assay how the gene is expressed, the more copies of DNA the birds have in this region, the higher the expression is in the feathers.
Now, why do you think that there are these different alleles in the first place? And do they actually have any impact on how the birds fare in the wild?
It appears that they do. Interestingly, there is some evidence from ecological studies not done by us that the birds that have the checker pattern fare better in specific places. And those specific places, oddly enough, are urban environments. The natural habitats for these birds are rock cliffs. You can think of like the cliffs of Dover in the UK. They also live in deserts in the Middle East. That's one of their native habitats and part of their native range. But in urban habitats, there are obviously more recent introductions. And the birds with the checker pattern fare better reproductively. Their offspring fledge better from the nest. They survive to adulthood. Interestingly, the converse is true in some rural environments. So in harsh rural environments, such as the Faroe Islands, there's not much food available. The birds with the checker pattern don't fare as well. And one reason that may underlie this is that the checker birds are not as good at putting on fat over the winter as the bar birds. So this is a huge disadvantage in places that have really harsh winters. But in cities where there are French fries and other things available year-round, these checker birds do just fine. And again, in some cases, they will breed year-round, whereas the ones with the bar pattern will stop.
Mike Shapiro. You're listening to the eLife podcast. I'm Chris Smith, and we're talking to the authors of some of the leading papers published recently in eLife. Still to come, a breath of fresh air in our understanding of the problem that is lung fibrosis. First, Mycobacterium bovis, which causes bovine TB, is a close relative of the TB bacterium that causes disease in humans. And because it can infect us, cattle are rigorously monitored. The problem is that there's also an environmental reservoir of the infection. Wild animals, including badgers, can carry it and, some suspect, can transmit it to cows. For this reason, they're being called to cull badgers. But is this justified when there is potentially a vaccine we could use? Unfortunately, no one knows how effective that vaccine would be, and before we can use it, we need the EU to change the law. They'll only do that if there's adequate data to defend their decision. So are the demands feasible? Andrew Conlon from the University of Cambridge Vet School models disease dynamics.
For the past 20 years, the long-term strategy of the UK government for the control of bovine tuberculosis in cattle has been the development and use of an effective vaccine. Bovine tuberculosis, although primarily affecting cattle, is a disease that can affect anything warm and furry, including humans. However, it's also known to infect many wildlife species, most controversially in this country, the badger. And it's the controversy over the culling of badgers to control the spread of bovine TB, which was one of the main reasons that cattle vaccination has been seen as the final end game for control.
Why should developing a vaccine for an infectious disease be anything other than a good thing?
So vaccines are, as we know, the most efficient way of controlling infectious diseases because they don't just protect the individuals that we vaccinate, but they also reduce the rate at which the disease is spread in the population, so-called herd immunity effects. The problem with bovine TB is the only vaccine that we have, which is the same BCG vaccine that we use in humans, has limited efficacy. It's not quite clear if it can demonstrate herd immunity effects in populations, and more worryingly, it interferes with the basic diagnostic test that is currently the cornerstone of control. And in fact, legally, the definition of disease is reaction to this test, which means if we were to vaccinate animals at the moment with the BCG vaccine, they would test positive and then we would have to kill them.
What can we do about that?
We could try and develop a vaccine which doesn't cause the reaction with the skin test. However, the problem with that is almost every vaccine which has shown any useful efficacy is in some way related to or based upon BCG. The second thing we can do is we can replace the skin test. So the current test is based on basically boiling up large amounts of the bacteria to get a soup of proteins which we then inject into animals and see how strongly the animal reacts to them. If we could replace that test with a more defined test, which just uses proteins which we know are in the wild bovine tuberculosis bacterium, but not in the vaccine strain, then we could use the vaccine without interfering with the test and slaughter programs that we use at the moment.
So what's the question that you've been trying to address with this paper?
This paper came out of a call by the government to design field trials for the evaluation of BCG vaccine in cattle. This was prompted by a chain of events started by Brian May, who is not only the lead guitarist of Queen, but is now a very prominent animal rights campaigner, in particular with respect to badger conservation. He visited the EU and made a lot of noise about why we weren't using vaccination. I don't know if these were actually connected, but about a month later, there was a letter from the EU which set out what they would require the UK to show in order for there to be a change in legislation to allow the use of vaccination. What they said is that we would have to do field trials, we would have to demonstrate the efficacy of the vaccine on the farm, in the field, and we would also have to show that any replacement tests to the tuberculin test, so-called DIVA tests, that stands for differentiate infected from vaccinated animals. Those DIVA tests would also have to be validated at the same time under field conditions. As part of this, I had already developed some mathematical models which described the spread of bovine TB and the possible use of vaccination. So I was involved in the successful tender to design those field trials and show how large they would have to be in order to address all of the questions that the EU wanted to see satisfied.
And what factors have you taken into account when you've drawn up this model?
The very basic assumption is that bovine tuberculosis is an infectious disease. And we built a model which describes the transmission between cattle and also includes the possibility that infection comes in from other sources. And it also describes how testing is currently used in order to remove infectious animals from herds and how vaccination might interact with a testing regime.
Now, if you take all of the assumptions you've made and you take the predictions of your model, how realistic are the expectations and demands of the EU for the implementation of vaccination? Is this something we could do?
The model suggests that trials along the lines that the EU has suggested would be extremely challenging. They would have to be of a scale that is almost equivalent to doing a deployment of the vaccine at the national level. So to understand the basic question of whether the vaccine works or not, that's actually quite straightforward. It would take, the model suggests, about 100 herds. However, to address this specific question of showing that the vaccine actually reduces transmission, that would require much larger scales of the order of 800 to 1,000 hertz. And the reason for this is quite simple. It is that the models suggest that even if there was a very strong effect of the vaccine reducing transmission, we just wouldn't be able to measure that effect on a reasonable timescale of a field trial. So we'd only have around about 5 years to do a field trial, and because of the very slow transmission of bovine TB, there just won't be enough data to quantify that effect.
So is our answer to the EU then that this is just not practical?
We make a series of positive steps that could be done. So in particular, if this question about what effect the vaccine has on transmission is necessary to be asked, and actually the EU has good reasons for asking for this, because the big concern with introducing vaccination is that if you nobble the skin test and you start missing truly infected animals, but the vaccine doesn't work, you could actually see an increase in disease, even though there's a reduction in the apparent level of disease. So there's good reasons for it, but we would argue that a field trial is not the best way of doing it. In fact, we propose some simpler natural transmission studies, experimental studies under much more controlled settings, which can be used to address this specific question. And these are experiments we're already planning to do in India and Ethiopia in separately funded projects. But we would argue that if this question is important, and it is important, that is the more appropriate way to ask it than through field trials.
Well, it will be very interesting to hear what those trials reveal. Andrew Conlon there with also what's possibly the first reference we've ever had to a rock star in an eLife interview. Finally this month, scientists at Southampton University have some good news about the problem that is lung fibrosis, where patients' lungs become stiff and gas exchange is severely impaired. Prevailing wisdom was that this was caused by a build-up of excessive connective tissue, and indeed you do see that in end-stage disease. But by studying biopsy specimens from patients early in the disease course, Mark Jones has discovered that the structure of the collagen in their lungs is different, and he thinks he can intervene with drugs to retard the progression.
I'm a lung doctor, and one of the conditions that I see a lot of is lung fibrosis, and in particular a type called idiopathic pulmonary fibrosis. Unfortunately, we don't have very effective treatments, and one of the questions I really wanted to start out to address was to understand exactly what's happening at the time that fibrosis develops. So how does it start and what progresses over time? And the thing that we chose to focus on was collagen.
Now, if one looks at the lungs of a person diagnosed with fibrosis, what do you see, both macroscopically but also down a microscope?
We can look with a CT scan. We see that the bottoms of the lungs are scarred, so you see lots of little white dots on the scan which shouldn't be there, and there's just far too much protein there, and we think that that's extracellular matrix. If you look even closer down with a microscope, you see more and more of these changes, but we really don't understand why they start.
And when you look at the composition of those proteins that are building up, is it just more of what we should have there, or is the distribution and the different concentrations of the different components wrong?
So we don't really know. There's a concept that collagen, which is really thought to be the main building block of the lungs from a structure point of view, is increased over time. But one of the challenges is how you relate that to other components. We don't have a very good way of measuring and comparing.
So what did you actually do then? Who did you study, and how did you try and get to the bottom of what is actually causing this?
We wanted to look at collagen. It's quite complicated in the way that it can assemble. So you can start with an individual brick or collagen fibril, as we can call it, and each of those is around 1/1500th of a human hair. But when they then join together, they get bigger and bigger. And so when we want to look at it, one of the questions we can ask is, well, is there more collagen there? Or is actually how the collagens joining together changed? And one of the things you can look at there is cross-linking.
Because obviously these diseases are complicated, and what starts the process may not actually be where you end up, as in you could end up with lots of collagen there in someone who has end-stage disease, but that wasn't what caused the disease to start happening in the first place. So can you get a handle on that?
Exactly. So one of the ways that we can diagnose fibrosis is by taking a surgical lung biopsy. We were able to use the tissue which wasn't needed for diagnosis to actually look at the changes which had happened in the lungs at time of diagnosis. Normally, studies have looked at very much end-stage tissue, so we were very fortunate to be able to look at this tissue at time of diagnosis. What we found, actually the amount of collagen hadn't increased, but when we look at each collagen fibril, there seem to be fundamental differences. They're smaller, and the way that they join together seems to be more intricate, so they have more cross-links. One of the reasons that that's important is actually that can affect how stiff the lungs become. And we know that the stiffer an area is, the more likely fibrosis is to progress over time. And so the question then is, well, what's driving those changes?
That's interesting, isn't it? So the collagen, almost the recipe that's being used and the way it's being assembled has changed between a healthy lung or someone at the very beginning of this disease and someone who has got end-stage disease. It's not the overall amount of collagen, it's the way it's wired up.
Exactly. And that's a kind of a really important concept to think about, because actually when we then look at treatments for fibrosis, very often in the studies that we're doing before we're looking at trying a drug in patients, we need to think, well, what are the measures we need to look at? Very often we actually look at the amount of collagen as being proof that, yes, there's a possible drug and it's possibly might actually work in patients. What we saw was that actually that amount of collagen seems far less important, and what we really need to focus on is how is the collagen structure changed, because we think that that might actually be something which might have far more benefit from a treatment point of view.
Apart from finding that the strands are physically different in the sense they're a bit smaller and they are stiffer, how have you managed to unpick or untwist, if you like, these strands to work out why they are like that?
One of the kind of challenges when it comes to studying something like fibrosis is that we can look in the tissue, but we know that's just one time point, and really to then try and unpick what might be the underlying mechanisms of this, we need to be able to look in models where we can look at changes over time. And so we were able to use cells from patients and actually start to grow little areas of fibrosis, and we can look at the collagen over time. We were then able to work with pharmaceutical companies with drugs which targeted some of the changes that we identified, and we were able to then start to unpick how those changes might relate back to the patient.
So do you think this is almost a bit like a prion disease in the sense that you get an abnormal folding of a protein that then makes more of the same, and that once the collagen starts to abnormally behave, it then encourages more collagen to behave badly?
Exactly, yes. I think this type of feedback loop, which can self-perpetuate fibrosis, is definitely something that we saw from our data and other groups have shown.
And the pharmacology that you think you can throw at this, how does it work, and is it very effective?
We were able to look at the specific types of crosslinks that we thought were altered, and we can use a specific drug which is able to inhibit those crosslinks. And I think the studies that we did, it works very well, and we seem to get a resetting of fibrosis. But the next step is really to translate those treatments from our studies into human studies, and so the very first part of that, the phase 1 trials are in progress at the moment. So hopefully there is the potential for this to be of real benefit to patients in the future.
Well, that's great news indeed. Mark Jones there from the University of Southampton. And there we must leave it for this month. You've been listening to the eLife podcast, previous editions of which and full text transcripts, as well as the references and details on how to subscribe, are available on the Naked Scientist website at nakedscientist.com/eLife. I'll be back with another look inside eLife next month. Until then, though, thanks for listening. And from me, Chris Smith. Goodbye.
The eLife podcast from eLife, the open access journal for outstanding research in the life and biomedical sciences. Online at elifesciences.org.